Oh, I'm going the wrong way now. Okay, looks like we're ready to roll then. Anyway, I want to start off by thanking everybody for inviting me to come and talk to this group. It's been a very long time since I've done that. I think it was, I think it's probably been 15 years, so nice to be back. Today, I'm going to talk about seeing the trees for the forest. And my title looks a little different, but it's basically the same. Can whitebark pine persist into the future? And I think this is a particularly good topic right now because if you've been watching the news, you may know that whitebark pine was just recently listed as threatened under the Endangered Species Act just about 3 weeks ago. And so if you're familiar with this tree at all, you know that it's this really amazing tree in the high elevations, but that it's under really serious decline. It's got a lot of problems, a lot of threats. And so I think a lot of people are quite excited that it has finally, after about 20 years of trying, been listed. And I think a lot of that excitement comes from just how successful the Endangered Species Act has been. It's been pretty phenomenal if you look at the track record of recovery of species that have been listed, and not just bringing them back to a certain degree, but there's a number of them now that aren't listed anymore and are doing quite well. But I think unfortunately in these days we have a bit of a harder time recovering species because it's not as easy sometimes as going out and figuring out, oh, it's DDT that's causing thin eggshells or this or that, and alleviating that problem. We're now facing some really difficult threats like invasives, which are just devastating to ecosystems. We've got climate change, which is not something you can go out and deal with easily to recover a species. So that's one of the problems I think with whitebark pine is it's now listed, but what does this really mean for this tree? What can we actually do to turn around the decline? I think one of the things listing means is that potentially there'll be a little more money for restoration and recovery. To date, there hasn't been a lot of support, but I think more money may flow in this direction. And I think a lot of that will probably go into what's already being currently done, which is locating rust-resistant trees and then planting these out onto the landscape to try and get more trees back on the where they've declined. This is probably a good place for me to kind of back up a little and talk about the threats that actually this tree is facing. One of the big ones is an invasive pathogen that causes white pine blister rust. And this one has been affecting this tree for decades. And people have been working on finding resistance, getting seedlings that are resistant onto the landscape. And after decades of work, this has become a fairly successful program. But unfortunately, blister rust is not the only threat that this tree is under. We have climate change, we have different disturbance patterns. And so one of the questions that comes up a lot is if we just follow the current restoration practices we're doing, planting rust-resistant trees, is that going to be enough? And I would argue no. And so I'm going to talk a lot today about some of the other threats and what we might need to be doing and looking at to try and deal with the other threats this tree is facing. Ecologists have this term, and I think economists and a number of other people use it as well. It's called a wicked problem. And a wicked problem is one where you have not just one problem, but you have several, each of which are very difficult to deal with. And that's what we have with whitebark pine. It's definitely a wicked problem. I like to think of whitebark pine as sort of a three-legged stool, okay? And that we have a threat chewing away at the three legs of that stool. And we know that stools have to have all three legs to persist. If even one leg goes, so does the whole stool, and that's true with whitebark pine. And so we actually have a situation where we have some serious problems, and every single one is going to have to be dealt with. And that makes this a really tough challenge. So what are these legs? These are the ones that I've listed here. In blue are the various threats. And so of course we have this invasive pathogen that causes white pine blister rust. It's a very serious threat. Some areas of the trees range, we've already lost 90 to 98% of the trees. So it is a huge threat. We also have two more big threats though, two more things that I think of as chewing away at the legs of this tree caused by climate change. And one of these is altered disturbance. So things are hotter, they're drier. We're starting to see more fires. We're seeing bigger fires, more severe fires. And that means we're seeing a lot more fire more often in the high elevations. And when you're trying to save the last remaining large seed-bearing trees that are up there, the last thing you want is to get fire. And the same thing if you're trying to get natural regeneration or these rust-resistant seedlings to establish, fire will take these out very, very easily. Another altered disturbance that's causing a lot of damage to whitebark pine is increases in bark beetles, and primarily mountain pine beetle. It used to be that it was too cold for these beetles to really persist in the high elevations, and so whitebark pine was pretty much protected. The only time it got up there was in periods of warm drought, like the Dust Bowl and some other periods. It killed a few trees, and then once things went back to normal and it got cold in the high elevations, the beetles were pushed off the mountains. Now, with warming, it's warm enough for the beetles to persist in these high elevation sites. And now they're basically residents because we're not going back to normal. It's continued warm and dry. And because of this, we've seen millions and millions of acres of trees, whitebark pines, killed by these beetles. The third leg is another part of climate change, and that's altered precipitation and temperature. It's hotter, it's drier, we have less snowpack, the snowpack is not lasting as long. We have longer growing seasons, which means there's more demand for water, but in many cases, we're getting the same amount or less. And so that means that this can stress the survival of these big old veterans. These are the ones that are needed to provide seed sources. They're also trees that have been well established, but there's really only so much they can take. So we're seeing greater mortality of some of these large trees. But probably just as serious or even more serious is that when you have drier and more variable climate, you have difficulty regenerating trees. Little conifers take 3 or 4 years of certain conditions in order to establish, be able to get a good root system established. And if you don't have that, they don't tend to survive. And so as we see this change in climate and the high elevation, we may begin to see big reductions in new trees replacing those that are lost. So these are 3 pretty big challenges. And so how do we address these? Well, in each case, the answer lies in understanding the tree's genetics, because it really comes down to adaptation. And adaptation is, I think, one of the most abused terms out there. I think a lot of people use it in a lot of incorrect ways. A tree that's standing out there in the forest right now can't adapt. It is what it is. It has its genome, its genetics. Adaptation is genetic change over time. okay, due to natural selection. And so if we're going to see solutions to these three problems that this tree is facing, it's really going to come down to the adaptation potential of these trees. And I thought I'd just throw this little kind of example of natural selection up here to show how adaptation moves. I think everybody in this audience knows how this works, but I found that this is a good way to kind of lay down the thinking of other things I'm going to talk about. And this is an example of an organism having to make quick change adaptation to changes in the environment. And it's a really famous example. I think it's in a lot of the ecology texts, but it comes from studies that were done quite some time ago. There was one in Eastern US, one in England, in areas where they had these big coal burning plants, and it was before emissions were really controlled. And so these plants would just spew forth tons and tons of black smoke, and the surrounding towns and forests, everything was basically black or gray because of all the soot. And there were these white trees, kind of look like birch, that had these white moths that had evolved over time to be very camouflaged to hang out on that white bark. But once that soot was in the air, these trees became covered, became very dark, and it caused these moths to be very apparent. They just stood out. They weren't camouflaged anymore. And that meant when predators came along, they were able to see these and they gobbled them up. Luckily, there was some genetic variation, some diversity in these populations for these dark forms, which you can see here. And those managed to escape the predators because they weren't seen. And in just a few generations, there was a switch over to almost all dark moths because the genetic variants that had the white had been almost wiped out. And those that were dark were beginning to reproduce and pass on those genes. Interestingly, once there were emission controls and that soot was controlled, the trees turned white again, and we had a switch back to all these white moths, because at that time, then the gray moths were very apparent and the predators ate all of those. But kind of the take-home message here is this population of moths may not even have survived if it hadn't had that genetic diversity to allow it to respond or to adapt to this change. And so we can think about that in the context of what is happening with whitebark pine. I like to tell people that genetic diversity is adaptation's toolkit, okay? And so think of it, if you have this big problem that comes up, something you haven't dealt with before, and this is your toolkit, and it looks like you've got something like 3, maybe 4 tools in there. What's the chance that you're going to have the right tool to deal with that problem? It's pretty low, okay? But what if you have this toolkit? The chances are you're going to maybe have not only a tool, but several tools to be able to deal with that problem. And so you can think of genetic diversity in the same way. If an organism has very little genetic diversity, the likelihood it has what it takes to adapt to change is low. But if it has a lot, the potential is there. And so we can use that understanding of genetic diversity to deal with these threats. And in fact, that's something that has already been done with blister rust. So what has been done over the decades with blister rust to try and find resistance is to go out to forests that have been really exposed to this pathogen for a long time, have had high levels of infection, and look for trees that aren't infected. Because after that long of a period of time and that much exposure, the likelihood that they're not infected by chance is pretty low. So they probably have genetic resistance. And so people climb these trees, collect seeds, they screen them by exposing them to the pathogens. And then there's those that don't show infection, those are outplanted. And so this approach of looking for genetic diversity that allows these organisms to persist in the face of a threat is something that's already being done with whitebark pine, especially with blister rust. But what about these other threats that are threatening the other legs of that stool, namely the bark beetles and the climate? Well, what we've been doing is trying to learn from post-bark beetle outbreak forests. And so instead of looking for forests that have been really slammed by blister rust, we go out into forests that have been really hit hard by the mountain pine beetle. And so we've looked mainly at forests that have experienced about 90 to 95% mortality of large trees. And then we've started looking at those few survivors that are out there. If you think about it, it's much like exposure to blister rust. When these beetles were in high populations in these forests, after a while, there were billions of them flying around. They killed most of the trees. They were running out of food, and they really had almost nowhere to go. But for some reason, they weren't going into these few trees. And so we wanted to see if there was some genetic basis to that survivorship, which means it could be passed on, which is what we need. That's adaptation. And we also wanted to see not only do they have something that's genetically heritable that has to do with beetle resistance, but we thought we might be able to take on kind of or kill two birds with one stone. We know that for most bark beetles, they respond to trees that are stressed out from drought and heat. And so we're thinking maybe these trees are drought tolerant. And so maybe that's why they weren't so susceptible to beetles. So if they're both resistant to beetles and drought tolerant, we can kind of address two of those legs at once. We were hoping. Something else we wanted to look at was what is the impact of these big outbreaks on genetic diversity? When you kill off huge numbers of a population and you only have a few remaining, one of the negative effects of something like that is that you may lose a huge amount of genetic diversity just due to chance. We call that a genetic bottleneck. And that's one of the things that's happened with so many of our endangered species is they've gotten down to a few individuals, they've lost most of the diversity, and then all you have to rebuild with is this undiverse population. So we were hopeful that we would find adaptation to beetles and drought in these forests, but we were also fearful that while that may help these trees in the short term, if we lost genetic diversity during these outbreaks in the long term, it may make it hard for this tree to deal with additional change. So we wanted to get a feeling for the diversity. So these are some of the questions that we looked at going in. We wanted to know if survivors were genetically different. And that would indicate that for whatever reason they're surviving, it could be passed on. But we also wanted to get an idea why they were surviving. So we wanted to look to see if it was due to the tree defensive chemistry. If you walk around in a conifer forest and you smell the resin or the needles, all that beautiful pine smell, those are actually kind of natural pesticides that trees produce to protect themselves. Still smells good, but these are defensive chemicals. So we wanted to see if they were different in the trees that survived. Maybe the beetles didn't recognize them, or maybe the survivors were producing more of some that were more toxic to the beetles, so they were avoided. We just wanted to see if there was a relationship there. We also wanted to see if survivorship was related to growth rate. Were they preferring trees that were growing fast or slow? And we wanted to see if survivorship was related to climate sensitivity. Were beetles, for example, picking off trees that were more sensitive to drought than others? So we located some forests. We've done 7 sites now, but I'm just going to talk about 3. One was Vipond Park, which, if you're familiar with this, it's high up in a plateau above the Big Hole River. About 95% of the big trees were killed during an outbreak there. Poverty Flat is another high plateau. It's in Idaho, a couple 100 kilometers away. Same elevation, xeric site, very, very similar. Both are very flat. We need a flat spot because we want to get rid of microsite differences like moisture and aspect, so that we can try and see if the beetles are choosing trees because of differences in the trees, not because of the conditions of where the trees were growing. And we also, and at Poverty Flat, it wasn't quite as bad of an outbreak. It was about 70% of the trees were killed. And then we added Lacey Creek, Montana, which was right across a valley from Vipond Park. It's only about 27 kilometers as the crow flies, not very far. But it was an area that was really the only spot in that whole area that had not been attacked by beetles. And so we were really curious as to what might have been different about that location. So at these sites, we went out and did some sampling. This is my experimental design, and I know it looks really weird, but I'll explain it. At each of the outbreak sites, we located 3 different types of trees. First, we located the bee or the beetle killed trees. Those were easy to find, obviously. And we measured the diameter for about 200 of those trees to figure out what diameter or size distribution that the beetles preferred. What size trees were they killing? Then we located the survivors, and these were trees of those same diameters that the beetles preferred that survived. So these were big trees. And then we located the G trees, and these we called general population just because we couldn't think of anything better. But they were mature trees, cone-bearing trees, but they were just a little too small for the beetles. So they were just a few centimeters smaller in diameter than what the beetles were going after. We included the general trees as a proxy for what we thought the population was like before the beetles showed up. Because they were a little too small, we would assume that this population was representative of the genetic makeup before the beetles. And so for like Vipond Park, where 95% of the trees were killed by beetles, we expected for the genetics, 95% of those trees would be of one type of group that was susceptible, and then about 5% would be identical to the survivors, if that makes sense. So for the bark beetle killed trees, the bee trees, about all we could do was do diameters, and then we collected cores. And from the cores, we could look to see how these trees might differ in their growth rates. And also, we're able to look at the sensitivity of each individual tree to climate, temperature, weather, different things, precipitation over their lifespan. For the survivors, we did the same thing. We got the cores, but we also measured resin flow because that is expected to be one of the top lines of defense for pines. And I'm actually not going to talk about resin flow anymore because out of a few hundred trees, we only had 4 that produced measurable resin. This tree really doesn't defend itself. Even the survivors weren't putting out resin. And we think this tree has really never had to evolve defenses against the beetle. So we quit looking at resin flow. We then went on to collect needles. From those, we could extract DNA, and we developed these genetic profiles using a method called ISSRs, which allows us to look at a lot of individual tree variability. And then we also took these punches out of the bark, removed the phloem, and then from that, we analyzed the chemical profiles for all of these trees. And then we did all of those same things for the generals. And so what did we find? And I'm going to talk today about some work we published in 2021, but we also have some similar papers out there that include this kind of work with lodgepole pine if you're interested, and hopefully in a few weeks on lodgepole pine. So for the three populations, though, that I've been talking about, let me walk you through what we found. And I'm going to start with the genetics. And what you're looking at here is called a principal components analysis. And it looks kind of confusing, but really it's not difficult. Each of these spots on here is a different tree. And if they're clustered close to each other, that means they're very similar in their genetic profiles. And the further apart they are, the more different. And so they're color coded as to groups. And then I've drawn the ellipticals around them to help kind of visualize where the groups are. So let me walk you through what we found for each of these groups. So PS are the poverty flat survivors, the Idaho site. And this is where we had about 70% mortality. So what we expected to find was about 30% overlap between the generals and the survivors. But what we found, if you look at the poverty flat survivors here in red, and then compare it with the poverty flat generals in green, was that they have almost no overlap. They're just really, really different, and much more different than we expected of our hypothesis that there should be something like 30% overlap. So there were some really big differences. And this is pretty unusual because you have to realize these trees are all growing together, right next to each other, all mixed up in the same forest. Yet they're genetically extremely distinct. And if we look at Vipon Park by the big hole, the blue triangles are here. These are the survivors. And then here in yellow are the generals. And here we've got quite a bit of overlap, which we would expect, but it's still more overlap than we expected because we only expected about 5 or 10% overlap. So again, they're different, but there's quite a bit shared. Here is the Lacey Creek site that we talked about, that the beetles just didn't show up, that there was no outbreak there, and we didn't know what made that one so different. It turns out that it's completely distinct from the Vipon Park survivors, which are only 27 kilometers away. There's no overlap at all, but they do overlap a bit with the Vipon Park generals. And they do overlap a bit with the Idaho beetles, which was a surprise. So this is a complicated story, and I'm sorry about that, but what we really have as a take home here is that we have some really genetically distinct groups, even within the same site. Some of it appears to be related to the ability to survive beetles, but there also appears to be something else going on that makes them different. And it also indicates that sites that are very close to each other can also be very different. And that indicates a pretty high level of local adaptation, which is something other people have found also in whitebark pine. So this is another way actually of looking at exactly the same genetic data. We run this through a program called Structure. And what it does is it tries to figure out from the profiles how many distinctly different genetic groups exist in that population. And in each of these three populations, the program detected 2 distinct groups, which are color-coded as green and red. And so there are definitely distinct groups. In poverty flat, the generals pretty much all came out in this red group, and most of the poverty flat survivors came out in the green group. And in White Pond, little more variability. Number of these trees have both green and red in the survivors. But again, most of the survivors are this group and most of the generals are of this group. Now in Lacey Creek, we have both groups again. We had no beetles though. And we struggle to figure out, okay, what does this mean? We've got potentially survivor and susceptible genotypes, but yet the beetles aren't killing these trees. Well, once we started looking at growth rates, we figured out that probably why there's no beetles at Lacey Creek is because of how they grow, which I'll talk about in just a second. Okay, so that brings me right to growth rates and climate sensitivity. And I don't know how many of you have had the opportunity to take tree cores, but it becomes addicting really quick. It's pretty magic. You core into a tree and then you pull out this core, which shows pretty much the entire autobiography for that tree. You can see its entire life and what it has been through, and it's pretty remarkable. If you look at this core here, each ring that you see is a different year in the life of that tree. And here you can see some of these are really wide. The tree was super happy, whoops, during that period. And then if you look here later in its life, or actually in this case earlier, it was having a hard time. It was barely growing at all. And so you can look at the overall growth rate of the tree, and you can also figure out what the date is for each of these rings and pair that up with the precipitation and temperature data for those years to see how that tree responds to drought, to wet years, all these different variables. So you can do a lot with these. Now, I don't expect you to read this table. I'm going to use it as a prop. Basically, in a nutshell, as we began to look at these trees, we realized really quick there were distinctly different growth rates in these different forests of these trees. So when we picked these sites, we intentionally tried to get forests that had, for the mature trees, close to the same diameters. So forests that look like most of the big trees were all the same age, what we call an even age stand. It just would have been easier to work with. And so they all looked very similar. But once we took the cores, we realized that in each case, there were two very different groups. There were those that were very fast growing and those that were really slow. So if you look, for example, up here for poverty flat, the dead ones are the beetle killed. And you can see there's what we're calling older and younger. So you have these ones pretty similar diameters. These are a little bigger. But if you look here, look at the difference in the mean age. These smaller, older ones are double the age of these younger ones. And that's not just in the beetle kill. If you look at the survivors, we have them pretty similar diameters, but look here at the difference. They're almost the same size, but look at, these are so much older than those. And that held for White Pond Park as well. And what was really kind of cool is to look at Lacey Creek. For those big trees we looked at there, they were pretty similar in diameter, but look at the age here. They weren't all that much bigger than these, but they were really old. 353 years old mean for the older and 200 for the younger. So these Lacey Creek trees were super, super slow growing compared to all the others. So take home, you can't judge the age of whitebark pine by their diameter. You have to take the tree cores. Something else about those different growth rates, that's typically genetically based. And so in each of those groups, we have very distinct growth rate differences, indicating different genetic basis. That's good because it means we've got a lot of genetic diversity there. Okay, so the next thing we did was to compare the kind of the lifelong growth rates of beetle-killed trees to those that survived. And for both those that were these older, slower growing ones, and for those that were younger and faster growing, it tended to be that the survivors tended to grow slower over their entire lives than the ones that the beetles killed. When we did the statistics, it was significant. The beetles were choosing the trees that were the fastest growing, and they were avoiding those that were the slowest. Something else we can see here, though, is that you can see over time, as we get to the late 30s into 1940, in each site, we see an uptick in growth in all of them. And so as we see more and more warming occurring in these high elevations, all of the trees are beginning to grow a bit faster. And something else that's notable is these generals, which are the younger kind of replacement trees, are the fastest growing of all of them at both of the sites. So that's a little bit of a concern, because if these are the replacement forests and beetles seem to like the faster growing trees, what does that mean for these trees when they reach a big enough size to be attacked by the beetles? Now let me finish this one thought. These are the growth curves for the Lacey Creek site. And you can see that they were growing rather slow over time. We don't have the beetle killed here because, of course, there weren't any. But something that's notable here is the growth amount. It's right around about 0.005, and then finally increases to about 0.01 increment by the time you get down here. But that's super slow. The other two sites were all growing up at around 0.025 and even above that. And so these extremely slow growing trees, perhaps because they're growing so slow, may have been lucky and escaped the beetle. Something I hadn't mentioned before, and I should have up when I was showing the structure analysis, is that we did look at the genetic diversity of the generals and the survivors. And we were predicting that the survivors were going to have very low genetic diversity because they had undergone this genetic bottleneck. So remember, like at Poverty Flat, there was only about 30% of the big trees surviving, and at Vipond Park, only about 5%. So with that big reduction in numbers, we expected to have seen a big loss in genetic diversity. And then we had all those general trees, the replacement trees that were very numerous. We expected they would have pretty high diversity. We found exactly the opposite. Those big old trees had extremely high genetic diversity, which was really encouraging. But what was sort of depressing was those younger replacement trees had extremely low genetic diversity. And that's a bit of a concern, because if we start losing some of those remaining big trees, it may mean that the replacement trees don't have a lot of genetic diversity to deal with change in the future. We're not sure why they're so low. It might be that they all established under particular conditions that selected against a lot of the genotypes that were there, leaving behind only those few that could survive under those conditions. We're really not sure, but it's something we need to look at. And I'll talk a little bit more about how we might do that in a minute. So we also looked at climate sensitivity, and I'm not going to take you through the massive statistics we had to do for this, but I'll just distill it down to the fact that after looking at a whole lot of different temperature and precipitation and drought variables, different times of the year, for going back about 50 years for every tree, we found that there were two distinct groups of trees. There were those that were very sensitive to drought, and that if there were drought years, about 9 months of the year, they would show strong sensitivity and reduce their growth. Then we had group 2, which we called our insensitive trees. And they just sort of grew the same no matter what you threw at them, whether it was drought or not, they just grew fine. They either showed no response or they would show a slight response to March and December, which we think is related to reduced snowpack. This got us really excited at first because we thought, oh, maybe we found what the beetles are cluing in on. I bet they're taking out those that are sensitive to drought, and those that are not sensitive, they're probably the survivors. Unfortunately, that's not what showed up. We had about equal amount of sensitive and insensitive trees in beetle killed as well as survivors. So unfortunately, at least from our analysis, we didn't find a relationship there. Chemistry, I'm going to go through this really quick. These are heat maps that you're looking at. And what it does is for all these different chemicals, which you can see listed here, those are the rows. It shows their production by an individual tree, which is each of these little lines going vertically. And if it's really dark, it means that tree produces a lot of that chemical. And if it's really light, it doesn't. It produces very little or none. And this is another cluster analysis that takes the chemical profiles and puts the trees that are most similar together and then separates those that are the most distinct. And what we found is that we had really different chemical profiles by population. So in pink are all those from Vipont Park. They were completely different than those over here from from Poverty Flat. And even within this population, we had some differences. The dark magenta are the old, slow-growing survivors. And we have this big cluster here that was very similar. And then we have some of those scattered through these with the younger survivors, and then the pink are the generals. But what we found here was that there was no clear indication that any of these were conferring resistance to the beetles at this site. Over here, this is for Poverty Flat. Survivors mostly clustered together, except for a couple little generals here, the light color. And then over here, the generals clustered with a few survivors. This sort of fit our prediction that survivors would be chemically different, generals would be more susceptible. And so at the site, maybe there is a signal. Overall, and we've looked at a couple more populations now, we haven't found any chemical signal that holds throughout any population. It does appear that there's genetic differences, driving differences in chemistry within populations, but we can't find a clear signal that it's related to trees surviving. So chemistry, again, just like sensitivity to climate, isn't giving us the signal we're looking for. So that was a lot of stuff all at once. So I just want to kind of summarize that. We found that trees that survive are genetically different, but we're not really sure what it is about their survivorship that's what the basis is. Terpenoids, the chemicals, may affect choice of trees within populations, but we really didn't find a universal profile across populations, so it may be completely related to those differences to something else. The one clear signal was that lifelong growth rate was a pretty good predictor of survival. So there's something about growth rate that affects whether trees are chosen or ignored by the beetles. We also found that old trees have higher genetic diversity. And so they might be the repositories or the kind of tool library of genetic diversity. And so it's probably of high importance for conservation for us to try and protect those big old trees. And as sources of diversity to try and restore these trees onto the landscape. Keeping that in mind, one of the things we're looking at is trying to better understand the genetic diversity within forests. Unfortunately, most of the time when people go out to assess diversity, they collect seeds from these big old trees or needles, and then what they find there is what they assume is the diversity for that entire forest. But from what we've seen, if we look at just even a cohort of younger trees down from those can be radically different in genetic diversity. And so now we're starting to compare what's in the big old trees to these other mature trees, to saplings, to seedlings, to see if indeed we're beginning to lose genetic diversity, or the diversity in each of these cohorts contributes as a whole. Because trees, once they reach a pollen and cone producing age, intermix with each other. And so these are providing pollen for these trees, and these cones are being pollinated by these. And so there may be more mixing and genetic diversity than we think just by looking at one cohort. But regardless, understanding the genetic diversity that remains in these forests is going to be key to understanding what kind of toolkit they have that may or may not allow them to persist. Anyway, with that, I'd like to conclude by acknowledging a lot of help. We didn't have a lot of money, but we had a lot of enthusiastic people working on this. And we have some ongoing work right now with BLM looking at some other sites in Idaho that are very different, very mesic. And we hope to get a bigger picture of what's happening across kind of the population landscape of this tree. And so with that, I'd be happy to end and answer any questions you might have.
Awesome, thank you. That was super interesting.
Cool, I hope I didn't talk too fast.
No, not at all. It's not for me. So I can't speak for everyone.
It's very different lecturing on Zoom than it is when you have people with you.
Yeah, you can't see anyone's face or if they're following what you're saying. So going back to the start of your presentation, Jackie was asking about fire, and I think we hear that subalpine fir can outcompete a whitebark pine successionally, and they're wondering if even low intensity fires that take out competing subalpine firs would be bad for whitebark pine in your view.
It depends. This is really a trade-off, because some of these forests need to burn to thin them out to allow whitebark to persist. But unfortunately, the amount of heat you need to take out the subalpine fir is also going to take out whitebark pine regeneration. And so I think it depends where and when you get the fires. There was a lot of push to introduce fire back to get more whitebark pine regenerating, but now I think there's a lot of fear that we're going to lose a lot to fire too. It's a double-edged sword. Some places we need it, but unfortunately the whitebarks burn as well.
Gotcha. Edward asked if there's any DNA that can be analyzed from the recently killed trees and give us kind of a genetic profile for these killed trees.
We have tried very hard to get DNA from these dead trees and dead needles, and we have not been able to get DNA. There are some people that said that they have been able to do it. It's a pretty complicated and expensive process. We haven't had the funding to do that, but I think the methodology is out there. And so I'm hoping we can ever get funding, or somebody will begin to do that, because then you can do direct comparisons. Mm-hmm. We were stuck using these slightly younger trees, which turn out to be different in other ways. So to do direct comparisons with beetle killed and big survivors would be the best, but we have not been able to get the DNA.
Yeah, interesting. I have kind of a two-part question. So you talked about how these trees don't put out sap or seem to have like chemical defenses against the beetles, and but the factor that was common among the survivors was this slow growth rate. So is your like hypothesis at this point that it's something about the like morphology of the cells in a slow growing tree that makes them less appealing to beetles? Like there's just too much cellulose in there or something?
We don't think it's the cellulose, but we think
Okay.
it is wood anatomy, or wood anatomy may play an important role. Um, so xylem are these tubes of that basically go up and down the tree. That's what most of the interior of the wood is, and that's the conductive tissue for water. Fast growing trees make big fat tubes. Slow growing trees make little tiny tubes. And so if you think of those as they have to pull water up by capillary action. So the smaller the tube, the easier it is to suck water up that. If you think about these little tubes we use to get blood out of your finger, they just suck it right up. You take a milkshake straw, which is big and fat, it's hard to get anything up that. And the same thing works that way in a tree. The bigger those tubes are, the harder it is during drought for a tree to pull water up to the crown. So we think slower growing trees, because they have smaller tubes, have less demand for water. And that these trees may just be able to plug through just about anything. Fast growing trees don't have also the carbon to invest in defenses, although these trees don't seem to defend much. But we think it might be due to some of that. But we're also starting to look at something most people don't look at, and that's non-structural carbons. Beetles are not just picking trees because they're either defensive or not. They want the easy ones that don't kill them, but they have to make good choices for food. And we think that they're going after some of these carbon sources, not cellulose, but other carbon sources like sugars and carbs that'll be good food. And they can pick up those signals. The defenses kind of made sense, except what's really funny is a lot of people are finding this thing that with even lodgepole, the beetles are ignoring a lot of those defensive chemicals. It's not as strong a thing as people thought in the past. There's something else going on and we've just been missing it. Just haven't quite figured out what it might be.
Okay, that's great. That brings up the second part. I was hoping you could talk just a bit about the basics of pine bark ecology. And so obviously wood doesn't seem like the most appealing food source. So like how do they work with these fungals to actually digest
Yeah,
wood?
yeah. That's really good, because if you think insects are animals, we're animals, believe it or not, we have a lot in common with them. And that is that we need and they need almost exactly the same nutrients in the same proportions. And if we tried to get what we need from chewing and eating wood, we would starve to death pretty fast. And so would beetles. And so these beetles go through this whole battle with trees to get through defenses and get in there just to get to some place where they're going to starve to death. And so what they do to get around the fact that wood is a terrible food source is that they carry these fungi with them. And a lot of people here, I'm sure, have seen that, these blue stain wood that you see people make all the furniture out. It's really pretty. The blue stain is from those fungi. So they carry these fungi into the tree. They're in these little pockets called mycangia. And the fungi grow deep into the tree, all the way down to the heartwood, and they gather up all the nitrogen and phosphorus, all those things the beetles need to develop. And then they pump it back out literally to under the bark where the beetles are feeding. And so it's like getting this assembly line of food just brought to the larvae so that they get the nitrogen phosphorus they need to build a body. And so that's how they're able to live in wood. And we think that possibly they're picking trees by the scent of which trees are going to feed their fungus best, so that that in turn will feed them the best. And so we're starting to look at some of these other cues, and that's all linked in again to those non-structural carbons and so forth.
It's a pretty remarkable skill to have, even if we don't always appreciate the result of killing a whitebark pine.
Yeah.
Can
The
appreciate
beetles
that.
are pretty amazing.
Yeah.
They do crazy stuff, you know. They eat tree toxins and turn them into their own pheromones to attack the tree. They do a lot of amazing things.
Great. And there's some comments in the chat for you too. Maybe I'll just give you a chance to read them at the end, but we'll just keep going with questions. Beth asked if there's any research on how to control blister rust, and I think I know the answer to this for you, but do you recommend the pheromone patches applied on whitebark pine?
The research that's being done on blister rust is mainly to find blister rust resistant trees. But there is some other work that, in fact, my PhD student, Lorinda Bullington, has done, where there's these fungi and bacteria that naturally live inside the needles of pines. And she's isolated these and looked to see if any of those can provide protection from blister rust resistance. And so she's found that there's a number of them that interact with the tree to produce a type of resistance. One is that they induce some of the defense chemicals that prevent the fungus from infecting the tree. And then others create some other kind of a change in the needles where there's more resistance. So the idea is if you can find these fungi and bacteria that are benign, they don't hurt the tree, you can spray a tree, and once they're infected with these benign fungi, then they won't get infected. And so she's been working on that for a while. She's also looking at mycorrhizal fungi in the roots and whether they also influence infection, and indeed some of them do. And she's busily analyzing her data right now, and hopefully in a few months we'll have the results of that. But that's another kind of way they might be able to protect trees, is just infect them with these benign fungi that can then protect them from infection.
That's really cool.
Yeah, and they're naturally occurring. These are not alien or exotic fungi we're bringing in. These are things that are out there naturally.
Greg had a question that I accidentally stole and asked already. So sorry about that, Greg. And I think I got to any of the questions. Anyone else can feel free to unmute themselves too at this point if you want to ask Diana something directly. But yeah, it seems like everyone enjoyed it. Thank you so much for joining us. I'll leave the call open. Oh,
pheromone
yes.
patches.
Pheromone patches, yeah.
I guess they're better than nothing. They're when you have really high outbreak populations, they really don't work. But right now, because the beetle has kind of collapsed down, they are pretty protective when you have lower populations of beetles. And so those blister rust resistant trees, at least, and some of these survivors, it couldn't hurt. They're not cheap, but if we can keep some of these big old trees out there on the landscape, you know, I think it's not a bad idea at all.
Awesome. Well, thank you everyone for joining us tonight, and thank you very much to Diana for your work, and look forward to seeing that paper hopefully in a couple of weeks from you. And yeah, it was great to learn. I'll leave the call open for a little bit because Diana, there was a kind of longer message from Wendy that just a comment for you, but you could have a chance to read that. Yeah, I hope everyone has a great night, and stay warm out there and enjoy your winter.
All right, thank you.